By seed quality, we mean the level or degree of their uh, suitability for a particular purpose producing crops. The seed testing laboratory is hub of seed. Okay, so above seed and what here quality control. The seed testing services are required for the from time to time gain information regarding the planting values of seed lots. So seed testing is possible for all these who produce, sell and use seeds. So objectives, what are the objectives here you can see. So this is the some objectives are there for the seed testing. What are they here? Seed testing is required to achieve the following objectives for minimize the risk of planting low quality seeds. That is first one to identify the quality problem and their probable cases to determine their quality and that is the suitability for planting. Third objective to determine the need for drying and processing and specific procedure that should be used. So determine if the seed meet established quality standards or labeling specifications. So fifth objective establish the quality and provide a basis for price and consumer discrimination among the lots in the market. So, so brief history of seed testing. So the international organized seed testing started more than the 100 years ago to avoid mm, unprincipled practices prevalent in the seed trade during the 19th century. The first lab for seed testing was established in Trond in Saxon, Germany in 1869 under the direction of Frederick Nobel. A few years in 1879, a seed testing laboratory was opened in the Copenhagen, Denmark, under the direction of Emil Holst. At the beginning of the 20th century, about the 130 seed testing stations were operating in Europe. In the United States, the first seed testing laboratory was opened in 1876. In India, the seed testing was station is established in 1961. So, International Seed Testing Association. This is that one here as a seed testing developed. Seed testing developed that it is then accessible for the establishment of common methods for testing the world security uniformity in the evaluation and test results. This leads to the information regarding the International Seed Testing Association in 1924. The ISTA is the rule to test the seed the following by the member countries. It is carrying out the seed testing work. The introduction of the International Seed Analysis Certification, widely used in the international seed trade, is another important achievement. So, AOSA means Association of Official Seed Agencies. The need for the standardization of seed testing method to lead the formation of organization in 1908, then known as Association of Official Seed Agent Analysts of North America. The basic objectives of AOSA are to develop and adopt the published rules for testing seeds to be encourage research in the seed technology. Importance. The importance of seed testing was realized more than 100 years ago for assumed planting the values. The alteration of vegetable seeds by stones, dust, which were packed in the same parts world, particularly in the Europe. Seed testing has been developed to aid the agriculture to avoid some of the hazards of crop production by finishing the needed information regarding the quality attributes, the purity, moisture, germination, vigorous, and health. The quality control of seed depends on the different seed testing period, which will determine the purity, moisture, germination, vigor, and health. So, quality control of seed depends on the different seed testing protocol, which is determine the germinance of the cultivar. So, standard seed testing procedure for the evaluation of seed was developed by ISTA, is the International Seed Testing Association, that is the labor obligated and obligatory on the part of the seed analysis to follow the particular by FA and ISTA. Role of the seed testing laboratories. What are the rules for the seed testing laboratory to maintain the some regulations, right? So what are the regulations we are to discuss here? So seed testing laboratories are essential organization in seed certification and seed quality control program. The main objective is to serve the producer, the consumer, and the seed industry by providing nutrition and seed quality. So, mm, So seed testing. So seed sampling, the principles and procedures. So seed sampling is the aim to obtain a sample as is required size and consisting of same components as the whole lots of seeds. The quality of seed testing is the laboratory too small compared with their size of the seed lot which is insured to present. So every effort must be made to ensure that the samples sent to the seed testing laboratory accurately represent the seed lot in the question objectives. The objective of sampling is to obtain in the representative sample for the size suitable for the test. When the sample is originates from the seed lot, it is expected that the result reflects the average quality of the seed lot. Seed sampling, the procedure of the obtaining the suitable fraction of the seed lot under the investigation by taking small portions at random from different positions of the lot and combining them. Seed lot is a specified quantity of the seed physiologically identifiable in the respect of which is the seed test certificate can be issued. Types of seed sampling, the primary sample, composite sample, and the submitted sample, working sample. These are the some sample types here. The primary sample means small portion of taken from one point in the lot. Composite soil, it is formed by combining and mixing all primary sample taken from the lot. Submitted sample. A sample is submitted to seed testing laboratory. The size of the submitted sample is specified in the seed testing. So working sample, the subsample taken from the submitted sample in the laboratory on which is the quality test is made. So requirement of sampling, uniformity of seed lot. Only the homogeneous seed lot is considered for the sampling. Heterogeneous seed lot is unsuitable for the sampling. Size or weight of the seed lot. The minimum limits of the seed lot in the 20 tons for the large seeded species, that is the 10 tons for the small selected or seeded species. So sampling device, sticker or sleeve type dryer, bag dryer. So stick or sleeve type tire is consists of hollow brass tube inside the closely finishing outer shell or sleeves which is made by solid point end. The tube is sleeve has open slot in their shell. So in the walls when it is tube to turn until the slots in the tube and sleeve are in the line. So seeds can be flow into the cavity of the tube and when the tube is giving the half turn opening and closed. The tubes vary in the length and diameter and are made with uh, the or without the patterning of the different kinds of seeds. So here, the next one is the bin sampler. The bin sampler was constructed on the sampler principle as big as dryer, but the much higher, running up to the length and 38 mm in the diameter with 6 or 10 tons. These samplers are used for drawing the sample from the lots stones stored in the bins. Rope dryer. So this dryer is made in the different dimension to shoot various kinds of seeds. It is pointed to long enough to reach the center of the bag with an oval hole near the pointed end. The total length of the instrument should be approximately 500 mm, including the handle of the about 100 mm and point of 60 mm, leaving about 130 mm to penetrate into the bag. With the tube is about to 14 mm, but for the close and similar seed, 10 mm in the different the sufficient. The note tire is suitable for sampling seed in the bags, but not in the bins. Miscellaneous sample. This includes sampler, palms, bucket, stapler, and swaying thread or pipe of cans. So selling drivers, sample bags, replacement, labeled and sealing top. So the headlamps is scissor, fish fight, and flashlight and packing the material, stationery and stamps, etc. A lot is consumed. So general principle of sampling. The sampling should be carried out only in the person trained and experienced in the seed sampling and employed by the different organisms. Right? So different organisms which here I'm explaining the order. So seed lot shall be arranged that each individual container or the part of the lot is conveniently accessible upon the priority. So the size of the seed it should be not exceed maximum seed lot size here. So other thing Being equal, the large sample is more representative of the lot, that is the small sample. Moreover, if there is the choice is to gather to reduce the sample before the seedling were 4 degrees. For the seed lot in the bag or similar quantity. So number of containers, sampling in intensity. So brief history of the seed testing. So small container, when the seed is small contains, such as tins, cans, that is very useful. Other containers, procedure, preceding lines in the compare use seed, any other kind of the container or from stream of seed. Lot size, name of the primary seed sampler, here UP up to 5,000. So 500 kgs to 3,000 kgs. So 3,000, 1 kg to 20,000. One primary sample was such 50,000. So while taking the sampler from the machine, saved and cotton bags, a few stitches was the weight of the sampler derived from the has been. So sampling the seed testing laboratory, the seed sampling received in the laboratory are the requirements is to be mm, it is obtained to avoid the working sampler of trying carrying out weights. So mechanical divider method, random cup method. So mechanical derived method here. This is method is suitable for the all kinds of second and expert. So when you are submitting the sampler smaller than the prescribed weight is received in the seed. So laboratory, the sampler may be notified according and analysis within the LME. So the sampler submitted weight only, and it is not in the ordinance, the size and prescribed. So random cup method. This method is the particularly suitable for the seeds since is required working a sample up to 10 grams provided for then this one is So modified halving method, the aperture is comprise the try to. So after planar mixing, the seed is poured evenly over the grade. So the hand having method, this method is the restricted for the following flower genera. The chiefly, so here, so each of the four portions is halved again, giving the eight portions which is should be arranged in the rows for the four. Subsequently, the alternate portion are combined and retained. The process is repeated until the sampling of the requirement. So duplicate sampling. So in order to achieve the greater accuracy, the sampling can be done in the duplicate to um, instead of the one. So independent working sample are taken from each submitted sample. So each working sample is half size of the prescribed standard the size. So duplicate sample are analyzed for the different analysis that is the result and compared with the only the when go of both analysis that will much which are other. So next one essential requirement and supplies for the germination test. So these are the very essential requirements are present. So what are the essential requirements? Here is shown to. So these are the some requirements are there. Seed germination, what here the seed germination are the essential is requirement for the germination. Testing for maintaining the specific condition of temperature, relative humidity and light. The seed germination are generally of two types of um, only that will be the germinate. The combination, the cabinet and seed germinators are essential under the situations where the various pest kinds of seeds that is required, the different seeds sets of the conditions are being the 100 in the laboratory. The number of species of the primary germinators is required by the laboratory will depend on the number of seed sampler and species being analyzed by the laboratory. The seed test laboratories that is handle the large number of seeds. So the seed testing is laboratories that maintain only few or 2 or 3 sets of temperature conditions. The walk-in germinators that is performed such as the germinations are more useful for conducting the germination sets. The sand media, it is require large germination seeds. The counting device includes the counting board and automatic seed counter and vacuum seed counter. The seeds device are required for the to avoid the germination testing by minimize the time spent on the planting of the seeds as well as the to provide proper spacing of the seed to germination substrate. So counting board are suitable for the medium. So counting boards are suitable for medium and bold size seeds, while the vacuum counter can be used for only small size seeds. In the absence of counting device, the work may be accomplished manually. Other equipment is required for germination of testing fluid in the refrigerator, scarifier, hot water both, including forceps, spatula, germination boxes, and plates, plastic plates around that is stainless plates, miscellaneous sampler, glassware, and chemicals. Germination proper paper. In addition, the laboratory may also require more glassware such as petri dish, beakers, funnel, measuring cylinder, measuring cloth, rubber bands, and tubes. So certain chemicals like potassium nitrate and thiosin, gibberellic acid, and tetrazolium. There is a chloride for a specific purpose. Voltage stabilizer are the required for supply. Done. So the voltage is suitable, essential for the fast generators, air conditions, and the refrigerators. The methods to improve the germination. The hard seeds, soaking, and mechanical scarification, acid scarification, inhibitory substances, mm, distinction of seeds, free chili. These are some methods to break or to improve the seed germination. Hard seeds. For the many species where the hard seed occur, some special treatment is essential. The treatment maybe is applied for the So prior to the commencement of the germination test or in the surprise, the treatment may be advisedly offered on-board seeds. It should be carried out the head seed maybe. Soaking. The seeds, there is a hard seeds. So mechanical certification, the careful persistent and other. Mhm. House level, BCBA, and Chinese course. Mm. So acid scarification. So treating with the concentrated sulfuric acid is effective with the some species. Digestion maybe rapidly or slowly 1 hour, but the seed should be examined every few minutes after digestion. Seeds must be thoroughly washed in the running tap water before the germination test is concentrated. In the case of Oryza sativa, scarification may be performed by the soaking the seed in one normal nitric acid for 24 hours, inhibitory substances. So normally occurring that the substances in the pericarp or seed coat which act as inhibitory for the germination may be removed by the washing the seed in the running water at the temperature of 25 degrees. So the germination of certain species is promoted by at the temperature. So removing the outer structure such as involucre of the bristle or the lemon or petal of the lamina or palea of certain piece poaceae. Disinfection of the seed, for example, for sample of the Arachis hypogaea and beta vulgaris, only a fungicide treatment may be applied before planting seed for germination. Then the seed lot is the chemical or percentage of the active ingredient. The method of treatment shall be reported on the certificate. Pre-chilling. So mm, in some seeds showing the that is the physiological dormancy, pre-chilling is required for the including the germination. So the replicate for the germination is the placed in the contact with the moist substratum and kept the low temperature for the initial period before they are removal to the temperature and in the So temperature between 3 to 5 degrees. So for the period and varying with the species from 7 days to 12 months. In some cases, it may be the necessary to extend so duration and the temperature should be reported on the analysis card. Pre-drying. The replicant for the germination should be heated at the temperature and not exceed 40 degrees with the free air circulation for the period of up to 7 days before they are placed under the prescribed germination conditions. Chemical treatment, potassium nitrate and gibberellic acid. So the germination substratum, so maybe the moisture solution or the potassium nitrate is included in the seed testing rules. So the substratum is the beginning of the test, but water is used for moisture. Uh, if uh, the use of this treatment should be noted on the analysis of certificate. So gibberellic acid. So Marston, the germination substratum with the 50 ppm solutions of the gibberellic acid, which can prepare by the dissolve 100 to 500 grams of gibberellic acid in the 1000 ml of water. Place the seed for the germination under the prescribed temperature condition. So, next one will be some evaluation of seed testing. Here, how we are to evaluate evaluation of germination test. So the germination test need to evaluate on the expiry of the germination period. So which varies according to the kind of the seeds. However, the seed analysis may be terminate the germination test on or the before the final count today or extended the test behind the period depending. So the evaluation germination test is seedling and seeds are categorized into normal seedlings, abnormal seedlings, dead seeds, and fresh hangers and minutes and odd seeds. It is may also be necessary to remove the seed coat and the separated cotyledons in order to examine the femule is the species where the essential structure still enclosed at the end of the test. Normal seedlings, it is necessary to separate normal seedlings which are the counted in the percentage germination from any abnormal seedlings to achieve uniformity, evaluating the normal seedlings. So seedlings which show the capacity for the continued development into normal plant when the grown in good quality soil under the favorable condition of water supply, temperature, and light. Seedlings possess all the following um, that is the essential structure where the tested on the artificial substrate. So, that is some. So, abnormal seedlings. There is abnormal seedlings are those uh, which do not show the capacity for the continued development into the normal plant. Hmm. When grown in good quality soil and under the favorable condition of water supply treatments, that is temperature and light. So the sealing with the following defect. shall be classed as abnormal. So damaged seedling, the seedlings with no cotyledons, seedlings with the construction splits and cracks, lesions with the effect that they continue conducting the tissues of epicotyle, hypocotyle, or root. So deformed seedling, the seedling with the weak or unbalanced development of the essential structures such as the spirally twisted or stunted plumule, hypocotyle or epicotyle, swollen shoots and stunted roots, split the plumules are coleoptile with the green leaf, watery and the glassy ceilings are white to further development after increase the cotyledons. So decayed seedling, the seedling with any of the essential structure is so diseased or destroyed, that the normal development is uh, prevented. So seedling shows that the cotyledon development from the micropyle or radical development from the part of the seed other than the micropyle. So here roots, that is the no roots in Avena and that is the triticum and that is the seasonal root only. So hypocotyl and epicotyl, hypocotyl short, thick and twisted are either curled over or watery. Epicotyl are stem with the construction the grain lesion and are open slit like in the infantile with the conducting tissue. Hypocotyl with the construction grainly lesions are open split likely to lesions or interface with the conducting tissues. Epicotyl or stem are that is the shorter and thick or twisted around the main axis. Uh, no terminal buds. There's the two shoot which are the short and weak are spindly. No primary leaves. Coleoptile. There is no green leaves. That is cotyledons, no cotyledons. One are evidence of damage on shoot apex. So purely developed leaf-like cotyledon allium with this definitive bend or knee that is enlarged with the hypocotyle. Physiological necrosis, gray in color, there is swallow and blackened. These are some ungerminated seeds here, hard seeds, fresh undergerminated seeds and dead seeds. These are the some germinate, right? So now coming to the another topic, that is the another lesson is there, that is the seed certification. So seed certification here.